Thank you very much for coming, Mike. So Mike is the co-founder and chief executive of Microbiotica, a biotech currently based at the Wellcome Trust Sanger Institute in Cambridgeshire. The company plans to transform personalised medicine by developing novel microbiome based diagnostics and therapeutics, and they have currently have programs in inflammatory bowel disease, immune-oncology, and treating Clostridium difficile. But we'll hear more about that shortly. Mike has 30 years experience in the biotech and pharma industry, has previously founded Crescendo Biologics and worked there as a CEO and CSO, and has also worked for many years at GSK, so has a really good experience across the board. And I believe you also support the life sciences charity LifeArc as a non-executive director. So if I could kick off, Mike, what made you decide to make the move from academia to working at GSK?
Actually, it was from academia to Welcome Biotech in the 80s. And so I started off, really, I would describe myself as a very commercial molecular biologist. And I was always interested in application. And I went the classic route first through, for example, John Scales' influenza group at Mill Hill. Then halfway into a genetic engineering institute in Leicester called the Leicester Biocentre. And then I was recruited into Welcome Biotech, which was a tremendously exciting company in the UK at the forefront of molecular biology in the industry, alongside Celltech and Searle. At that time, a lot was happening in California, but it was happening here too. And the first antibody was humanized at Welcome Biotech with Herman Waldman. There was a lot happening. I think really I was always stimulated by the excitement of applied molecular biology.
Okay. So when you moved to GSK from Welcome, I mean, what was the move for that?
This was actually, Welcome Biotech was the biotech company of the Wellcome Foundation, the drug company that Glaxo bought in '95 to make Glaxo Wellcome.
Okay.
So I stayed through a series of companies which were actually very, very different, starting with Welcome Biotech, a true biotech company, and then being absorbed into Welcome in the antiviral arena because I'd been a virologist. And I headed up efforts in papillomavirus and until Glaxo bought us and I started managing departments in target ID, target prosecution and genomics in Glaxo Wellcome. Until by around 2000 I was running the discovery division, about a third of UK research in Glaxo Wellcome under Clive Dix. And that's when then we merged with SB and I started having major international roles in SB.
Okay.
So GSK, not SB. That was a...
So obviously, very big pharma, GSK, so you then made the decision to found Crescendo. What was the thinking behind that?
The first thing is I think that the journey in GSK was not one thing. It did start with biotech, and I had one boss at one time, Mark Payton, who also was a CEO later, as were many of my colleagues, Melanie Lead, Roberta Solari, John Tite. And he said, if you start in a biotech, that makes a difference. And I think it did right through big pharma. But all that period was a tremendously exciting time because you could be an entrepreneurial scientist, though there were big differences from being in a small company. And by the time I left GSK, I was running a global group called Discovery Technology, which you could call as genomics and platform biology. It was 300 people on 5 sites. I'd also restarted the antibody pipeline with my colleague John Tite. So we did a lot, I learned a lot, and you do learn a lot in an excellent company like that. But there came a time when a few factors came together that told me it was time to leave. And I started thinking, I want to be a CSO in biotech because I thought this was the best job you could have. And I was actually searched out by a VC to start up Crescendo Biologics as CSO. We left the CEO job vacant and I stepped into it a few years later.
Okay. So how did you find that transition? Was it It's very smooth.
Very natural.
Yeah.
I mean, but I know that quite often investors are quite concerned about people who have been very successful in big organizations and maybe used to having a lot done for them, or operating within a framework, suddenly doing everything. But to be honest, it took me right back to the beginning.
Okay.
And also it got me from a level where I was managing 300 people and getting further from the science than I wished to getting right back down into it. Before we hired many people, I found myself helping to design very complex transgenic mice. It was quite scary, and but really refreshing. And so there was a bit of adaptation. And then I also went from delegating a lot to actually starting to micromanage deeply until I felt I had an organization I could trust.
Yeah.
And then I started delegating again. But I mean, it was fantastic.
Yeah.
Yeah, it was very stimulating. And as CEO, the level of accountability you have is total. There are not many jobs in big pharma where you have that. Maybe head of R&D and maybe the heads of therapeutic areas. But otherwise, that level of accountability, the need to do every job, you become immersed in it and it takes your life over.
So you've been now CEO of Microbiotica since 2016. I wonder, did you get some good learning experiences from Crescendo that you
Oh yes,
applied?
I think the second time is different. And I think actually I found that stepping out from the first one was easier to change than with the same old crew around me in the first one. I'd done Crescendo for 6 years, it had been very successful, and I felt that I could move on profitably and do something different, and I wanted to get into a new technology.
Mm-hmm.
But before I stepped into Microbiotica, I had a very interesting year and a half, which was one of the most stimulating of my whole career, if I may just tell you about that.
Yeah, of course.
First thing is I decided to move on from Crescendo, which is going really well, and I think it's a great company. Technology is working brilliantly. But I was quickly picked up by my old colleague from Welcome Biotech, Gordon Dugan, now Professor Gordon Dugan, FRS, who was head of pathogens at the Sanger Institute, who introduced me to Trevor Lawley, who he'd brought from Stanford to start the microbiome area.
Okay.
And they wanted some help to translate what was happening there. So actually I started off thinking, I'll help them out. I'll just help them out. And I embedded myself thinking, I'm not sure it's for me. But I just spent time there to understand it, help with an IP strategy, help shape it. And I started getting sucked in. And before I knew it, I'd had a few investors who wanted to support it. But it took a long time to spin it out, to get the license agreement, to get everything lined up, to spin it out finally by the end of 2016. And in that time, I got picked up by Imperial College, by Jonathan Weber, now Dean of Medicine at Imperial, to advise on translation with this amazing title of Translator in Residence at Imperial.
Yeah.
And I can speak a few languages, but I think actually Paolo and I we're the Mediterranean cohort, by the way. What I did then was try to understand the landscape in translation and provide some observations to the management of biomedicine across Imperial, which I did. And it gave me tremendous insight into the barriers and issues in translating for academics. And at the same time, I also advised about 70 PIs.
Okay.
From here to all the campuses. I was all over it for a year and a half or two, actually. And that was one of the most fun things I've ever done. I also joined the board of what is now LifeArc, which was going through a transition as it was getting the money in from licensing Keytruda. And I was also advising as a venture partner for what was called Rainbow Seed Fund.
Okay.
Yeah.
So you mentioned that you were looking at what was causing barriers to translation. I wonder if can you give any examples of some key things that came up at that time?
Yep. I think, firstly, I think that it's a very difficult job for a tech transfer group to address all the barriers because, this is a very big organization, Imperial College. I say this because we're almost in the heart of it. And so one of the biggest issues, I think, was lack of knowledge by many academics on what were the outlets for translation. And I mean not just commercialization, but even translation through academic routes. What would be a translational goal, a strategy? And I think a strategic way of thinking about that was sometimes absent. But it didn't take much in discussion with various people to get them to see and to suck that up. And I think one of the key things then was that they needed to be driving it. There needed to be more people who would push. I think somewhere like Cambridge has that, and I think that there it works a bit more organically. They're sitting next to each other on a high table in a college next to someone who founded a few companies.
Yeah.
And that helps quite a bit. So there was that need for strategic input, and what could you do? You could license a technology, you could form a spin-out, you needed patents. How did you build that? What did you have to do in your research to line it all up years before? And then other issues, the bottleneck of getting resources to, for example, to make small molecules or antibodies so that you could file a patent, and yet the pressure to publish at the same time. And so if you don't have those resources, you're under pressure to make a drug, small molecule or antibody, suboptimally, to file a patent suboptimally, where really what you need to do is have those resources quickly to get those assets. That translation actually then allows you to patent something and move on and publish. And so you're in this difficult situation. So those were some of the key barriers.
So do you think things are improving now? I mean, is there more of those kind of resources available now?
There are a few, and one of them actually is the organization I'm involved with now, LifeArc, which has expanded its brief. It was always as MRC technology helping translation everywhere it could. It's a translational charity. At the time, Imperial made very good use of the confidence in concept funds from the MRC as well. LifeArc is helping to fund 2 or 3 key areas where there was a gap. One other gap I saw at Imperial, and I was also on the MRC's translational funding panel, developmental pathway funding scheme. And I saw that there wasn't that much expertise for translation in the diagnostics biomarker area. There was a really lot of good science, but not many people who knew a path to commercializing it or translating it. Now, it is more difficult, but there was a gap in the UK anyway. And LifeArc has decided to partly fill that. It's built a diagnostics centre in Edinburgh and is trying to form a hub that gives know-how to academics and commercial entities to help beef up this area of translation in the UK. They're also doing that in antibacterials. And they've got I'm doing a big plug for LifeArc.
Yeah.
They're doing, they've got a philanthropic fund which is funding areas, for example, like gene therapy for rare diseases for philanthropic purposes.
Okay, so do they act a little bit like a tech transfer then, or?
Well, their main remit is around translation to benefit patients, but it's around translation. So there are now 3 funds. There's a philanthropic fund, a seed fund, and a full venture fund. There's direct funding, and there's research internally.
Okay.
Yeah.
Well, that's very interesting. I'm sure some people might want to
Maybe I could tell you about the microbiome now.
Yeah, of
if
course.
there's time.
Yeah. So yes, exactly. So there's been a lot of in the news about the microbiome in recent years. So I wonder, can you say a little bit about how does Microbiotica stand out from its competitors in the field?
Well, I'll maybe wrap it in with what I think are the barriers in the field, because there had been quite a lot of public investment in the microbiome, going back a decade with, in the US, the Human Microbiome Project, in the EU, MetaHit, and a Japanese equivalent. A lot of sequencing, shotgun sequencing of the gut, finding out that there were real correlations with changes in the gut microbiota in particular the gut, and disease. And it gradually became clear that the microorganisms that live in us, the microbiome, mainly bacteria in the gut, have an immense impact on every aspect of health and disease. And they're connected to the brain, to the immune system, to the metabolic system, the hormonal system, because the gut is so central, and the microbiome is reminding us of that. The gut bacteria can control or modulate all those processes. But there was a really big barrier, and the Sanger was able to put the resources in to address it under Trevor Lawley, and the barriers were, one, the gut bacteria were viewed as unculturable. People didn't think you could isolate more than 1 in 10 of them. And that was a problem that was cracked over many years by Trevor Lawley, and having done that, he could then collect species of gut bacteria, the biggest collection in the world, and he's put about 2/3 of the public gut bacterial species into the public domain with full reference genomes. And once you have those, then with shotgun sequencing, you can more accurately identify what patients have, and then you can find with precision what gut bacteria are doing what in patients. Now the company's taken it much further and industrialize it, so then we dwarf the public domain in reference genomes. We can isolate all the gut bacteria from any one person. That hasn't been done, and we've done it for 200 people, and that means we can get full definition in big cohorts of patients of the gut bacteria and correlate them with response. And I give you an example. In the response to checkpoint inhibitors, these are the drugs which are predicted in 2025, and Keytruda is one of them, and I was on the team that monetized that and brought in 1.3 billion for LifeArc. The market for checkpoint inhibitors is predicted to go to over $55 billion in 2025 because they address multiple cancers, they're broad spectrum. But the highest response rate is 40% for melanoma. In lung, it's more like 20%. It's very low. And it's really become clear that the gut microbiota are a key factor in determining response, but there's complete disagreement about which gut bacteria.
Okay. Yeah.
It's because two things. One, the studies have not been well powered or well conducted, and the microbiome profiling has been suboptimal. When you have a powerful microbiome profiling technology with machine learning to find consortia of bacteria that work together, then you can go to big patient groups and find what those signatures are. And we've been able to do that and get a consistent signal when the whole field is full of discord.
Yeah, well, you obviously are standing up above your peers because you managed to have an impressive partnering deal with Genentech.
They were watching what was happening in the microbiome field for years, wanting to enter it in the IBD area. They'd been watching what Trevor Lawley was doing at the Sanger, seeing the characteristic underpinning big science that the Sanger does. And they approached us soon after the company was formed. We had a due diligence visit in the 9th month with more people from Genentech visiting than we had in the company.
That's a good story.
And by the 18th month, we signed a deal worth on paper over $500 million. And it's been a fantastic relationship.
So they're a good partner to have.
Excellent partner, yeah.
Excellent. Yeah. I mean, you've obviously had a lot of experience in the industry. I mean, what do you think makes a good partner out of interest?
Well, I think there's a good partner that's different for each company. But for our company, I think it was absolute fundamentals of science. They felt that the microbiome had to go to a level of rigor that they saw in us. And we had a level of rigor that we didn't want to do a study with them that wasn't any good. We wouldn't have done it had we not been confident we could have got the results. And there was a complete meeting of the minds culturally on science first and business second. And I think, but there are different situations, there are different drivers for deals, so it's about getting the cultural fit, and we have a very good one. Yeah,
because of course some other people, sometimes think about going public to raise money. Is that something that you would consider, or are you?
Well, you can't just go public. I think we do have an ambition. We have a very big ambition in Microbiotica because we believe we have the leading platform for characterizing, profiling the microbiome at scale in humans, and we believe that's how it needs to be done first. before you decide what to do with the bacteria you find. You know? And so we think we should be as ambitious as our US counterparts. And so, you know, we are thinking of an IPO in the next 3 or slightly more years.
Okay.
Once we're in the clinic, we have ambitions to be in the clinic relatively quickly with assets we're progressing in ulcerative colitis and immuno-oncology.
Yeah, okay. So do you think, I mean, do you count microbiome therapeutics as being part of the advanced therapeutics?
I do. And I think this thing about advanced therapeutics or new modalities, and I've heard a number of people say, and actually the Genentech people did, they said, you know, it used to be small molecules, now our portfolio is almost all antibodies. But in a few years' time, they foresee it would be 10 different modalities. I've worked on small molecules, gene therapy, vaccines, recombinant proteins and antibodies. And Crescendo was something else again, and now this. And it took a very long time, actually, for antibodies to get through from 75, when monoclonals were invented at Cambridge, to humanization technology in the 90s, actually in the 80s, and then in 97 rituximab. I think this will be much quicker. I think there will be, and we're going to hear about another new modalities shortly.
Indeed.
I think new modalities will be essential. And they're more risky, but I think in some ways the most risky, I have to be careful what I say, but I think in some ways the most risky thing is small molecules. Okay. That's me being biased. I think, but I think that with the diversification of new modalities, there will be challenge, because antibodies has been such a rich seam.
Yeah.
And it's become plug and play in manufacturing, in development, in discovery. It won't quite be the same in the other new modalities. They'll be more diverse and more individual. But yes, the microbiome therapeutics, but microbiome therapeutics is many things. It's live bacterial therapeutics. It's phage targeting to remove some bacteria. It's metabolites from bacteria. It's many things.
So a bit broader than, say, cell or gene
No, but
therapy.
it's a whole new branch of biology. That's what's totally revolutionary about it. I mean, Genentech was driven first to understand the response of their biologics in terms of what the microbiome was doing. But then when you do that, you also find bacteria that could be co-therapeutics or therapies on their own. So I think there's a microbiome dimension that everyone has to take account of, actually. I think it's just a whole new dimension of biology. And our business model is to do that as well as the therapeutics. The biomarker side is you've got to understand what the microbiome is doing to your drug, and the other side is, and the bacteria can be a drug.
Okay. I think it's high time I open it up to the audience. So does anyone have any questions?
Thank you. May I ask, what does human modality mean in this context?
A modality. For me is a type of way of modulating, of targeting something in the body to make a therapeutic. So a small molecule is a modality, an antibody is a modality, antisense is a modality, gene therapy is a modality, and live bacterial therapeutics are a modality.
Yes, Mike, thanks for an excellent conversation. I'm curious because what you are doing in the try to push, let's say, the field in a different way with the translation, still is not like United States. What for you is the difference, despite the fact that you have a big amount of money? There is, to me, after 20 years in the United States, I was living there, still a mindset that is a little different on entrepreneurial front.
Yeah. I mean, Trevor and I, and with our investors, we're really driving the company aggressively, but it's noticeable that our competitors in Boston, of which there are several, tend to have 100 million in the bank. Right?
Kind of helps, yeah.
Although we do have the Genentech deal. But the money is a very important part of it, because you know, we want to expand into new therapeutic areas. And we have expanded quickly. I think it's easier to be more ambitious in the US. It's easier to take risks in new modalities. And I think US investors may support that in Europe, but less readily than in their own territory. I think if I compare our management with US management, I think we're just as ambitious. I don't know if that's true for everybody, but I'm sure Paolo is, and he's been in the US too. So I think it's more the environment we swim in.
Okay. Does anyone else have any questions? Gentleman in the middle.
Specifically thinking about actually altering the microbiome to make some kind of change you want to make, has an ideal modality already solidified out, or is it still happening?
Well, there are many approaches. I mean, there's also the engineered bacteria, but if you talk about putting in bacteria to change the balance, the pioneer there is fecal transplant. It's basically blended feces with a tube, right? And the amazing thing is it protects against recurrent C. difficile so well that it's been almost enabled by the FDA in the US, and there are public stool banks everywhere, although there was a death recently from a contamination. But in the main, it's safe. Later, it was found that it also cures a third of ulcerative colitis patients. It's absolutely remarkable. Actually, in that case, we got the clinical trial data set. We got the best data set because we have such a good platform, and we're mining the bacteria out of that engraft and correlate with therapy. And there's a very particular signature we found. So we would go to a pure product. Those pure products are beginning to come through, and some companies in the field have shown that you can deliver them orally. They die in air, so they have to be encapsulated, protected, and they can engraft long term. So that's one modality which is coming through. Already proof of concept from feces from the bottom side, but also freeze-dried feces in a capsule, less defined products, and beginning to get defined products in a capsule. So that modality is coming through, and I think it will be major. And then you've got companies already going towards the clinic to knock out bad bacteria with phage. There's different modalities here.
Thanks.
Can we imagine a space where we're not talking about therapeutics, we're talking about supplements?
Well, yes, I think people often ask about this, and of course there's a huge probiotics industry, and it's pretty big in Europe too, in the continent. And actually, a lot of that has led to more microbiome companies in continental countries than here, I would say. Although the thing is, when they're not clinically tested, they can't make medical claims, and then it's very difficult for companies like us to have a conversation with them. Some of them talk to us and say, can we share our bacterial collection or whatever, but it's very difficult for us to have a conversation when we can't see the data in the same terms. They haven't done clinical trials. The products are often powders of bacteria that probably don't survive all that well in air. Some of them just die, or they don't survive well in acid, but I'm sure some get through and have an effect. And I think that there are instances where they are having an effect, for example, in IBS, but I think it's going to be difficult to join up these industries. By the way, our third investor Saventure in Paris, is a complete specialist in the microbiome, and they believe that food, agriculture, and medicine will all link up with the microbiome. So they're invested in all of it.
I guess there might be a cost issue, because if you're going to have to do clinical trial type testing, then it may make it too expensive for the consumer.
It's the whole thing, really. We start off with precision analysis in clinical studies, then we get the bacteria out, then we manufacture them, we test them, then we go to clinical studies. Yes, it's a completely different thing.
Talking about science in a business environment, is there something which you did at Crescendo which you wouldn't do at Microbiotica again?
Is there something I did at Crescendo which I wouldn't? I mean, I've talked about, I think actually, I'm not sure I should say this, but I think that many companies have an original sin, right? Something that wasn't completely perfect at foundation, which always comes back.
Yeah.
And I'm not going to say what any of those things are. I think one of the things is you do need a good mixture of investors and a balance there. But I think more than things that I wouldn't do is things I learned that I applied better, more effectively second time round. And I was doing the CSO job as well, and that really stretched me in a crazy way. And so I'm able to be more strategic in this role.
Okay, perhaps one more question, and then we'll move on to the next speaker. Okay, gentleman in the middle.
Yeah, thanks. Just curious to know whether the effect of the microbiome on the therapeutic in terms of reducing its efficacy or enhancing its efficacy, would that create an issue? Would the sort of FDA require DDI type sort of investigation?
Well, every therapeutic we take now is affected by the microbiome. Almost everything. Some are very directly affected in that some drugs we take are eaten by gut bacteria. Right? And others, the gut bacteria are changing something, for example, an immune response in the gut or something else that mean that we should really be knowing in more cases. So the FDA are not yet in a position where they're going to act on any of this. It needs to be clarified an awful lot more. I mean, we're beginning to see in Microbiotica the ability to stratify responders to checkpoint inhibitors in melanoma in humans, and so we think we're going to be able to predict who responds. How will that be used? It's going to have to be very, very certain before you don't give the drug to people, right? But there are other instances where we think it can lead to side effects, and there you might be very careful.
Okay, well, thank you ever so much.
Pleasure. Thank you.